Welcome, everybody, back to the AWS Summit here in Washington, D.C., coming at you large and loud. I'm Kyle Dickinson, your neighborhood-friendly senior security solution architect that has a focus on threat detection and incident response. Time me on that one now, Aaron. I am with some friends here, and I just want to kind of steal a moment. Data is such a large conversation these days when it comes to technology, and especially when customers are using cloud-based technologies, specifically AWS. And my friends are here to talk about a specific type of data set, which to me seems to be very important with new research use cases. And I'm not going to continue on. I'll let my friends introduce themselves.
Sure. Well, thanks, Kyle, for having us. It's great to be here with you today. So my name is Erin Shu. I'm the global lead for AWS Open Data. My background is in genomics and also veterinary medicine.
Thank you, Erin. I'm Sujaya Srinivasan. I am the genomics technical lead for public sector. I'm also a bioinformatician, and I've been working with genomics for the last 15 years. So yeah, really excited to be here and talk to you, Erin, about open data and how we're helping researchers in genomics with our new Amazon Omics service, and how we can do this with open data.
So
Absolutely.
I want to take a step back real quick because you use a lot of very fancy words, and I'm a pretty simple person, but what is genomics? And you mentioned another awesome word. Could you just explain to us what these are?
Absolutely. So genomics is the study, kind of broadly, it is the study of what we call the blueprint of the cell, which is our DNA. Everyone knows that DNA is composed of 4 bases, A, C, T, and G. Me thinking that I got those right? Yes, I definitely got those 4 right. But what we do in genomics really is we want to understand how those DNA sequences are affecting the way we look, or our disease state, or how, for example, how the rate of mutation is being affected by the environment or by what we're eating. So when we talk about genomics, it's because we're looking at the entire genome of an organism. So that means every single base pair that one of their cells contains. And to do that, and just to give you a little bit of, I guess a level set, the amount of bases that you can see that the human genome composes, if you were going to print them out on a piece of paper, they would quite literally go from the bottom of the floor to the top of the ceiling of this expo hall.
3 billion bases.
Exactly. Just enormous, right? And
wow.
So you can imagine that the power of something like cloud with its scalable compute and analytics services is something that bioinformaticists, which is the kind of the informatic aspect of the study of biology, which genomics is a little sub-community of that, is incredibly appealing. Does that make sense?
It does. And so you specialize in genomics, you specialize in open data. Something tells me there's something really awesome that you're about to tell us about this specific data set for genomics.
Data sets. There's so many out there, and as Erin was just saying, I mean, the scale of the data is just huge. So if we were to just sequence one person's whole genome and do it at the, to get the appropriate quality, we're talking about potentially 80 gigabytes for one sample.
Wow.
And now we're talking, there's whole large-scale population sequencing efforts at the country levels, and we're sequencing literally millions of individuals. So think about, how big that could be.
Now, this doesn't seem like something that you would just have lying around.
Yeah, exactly. So security is a big aspect of it. And so, yeah, I'll let Erin talk about, what we have in the open data program related to genomics and the data sets, and why it's so important to have this as part of open data for researchers.
Yeah, I would love to. So, and forgive, our screen keeps to be glitching in and out. So really, this is a play to have you all very, very engaged on this session, because you don't know when it's going to go out. That all being said, like Sujaya mentioned, the privacy of the individuals that are sequenced is paramount. But actually, at the back of most clinical genomics pipelines, where people are saying, hey, okay, I've sequenced this individual, but I need to know, does this mutation, right? We all have a couple 100,000 mutations within our genes that don't do anything at all. So what is the clinical significance of a genetic mutation? Does it cause disease? Is it associated with disease in special populations of humans?
Okay.
So that information is typically encompassed in what we would consider open data. So data that are abstracted enough that you don't need to protect the privacy of the individuals. Usually it's at the population level. And most of these data have been aggregated by leading institutions who then want to make that data openly available to the world.
And so, you know, it kind of goes twofold with I say you don't just have this data lying around in a security sense, but also, you know, if you're a startup that wants to be inventive and also do research or like a research project, they're able to leverage these data sets or open data without having to generate it themselves, because that itself could be very time consuming.
Time consuming and expensive too.
Yeah.
Exactly. So, yeah, that's a great point. And so actually that leads a little bit into what my team does. So my team is a team of subject matter experts in various research domains. And what we do is we work with customers who say, hey, I want to share my data in the cloud. How do I do that knowing that I want to share my data with thousands or millions of people? Alternatively, we serve people who said, hey, I want to access this data in the cloud, and how do I leverage AWS services to use the data efficiently?
You used a particular word, efficiently, so I'm assuming that this data easily integrates with a lot of our different machine learning and AI services that we offer at AWS as well.
Sometimes. It's like, you saw Sujaya's face, she said, yeah, that's exactly.
Kind of. Ideally, yes, but there's a lot you need to do with the data, the way the way it's shared. I mean, it's great that we have all of this data that's being shared, but the format it's being shared in is not ideal to just like point SageMaker to it and just do something.
Not all the time. Yeah, and analysis ready data is definitely something that we talk a lot with our customers about, because sometimes what is analysis ready, what is cloud optimized, we don't actually have the answers to some of these questions for some research domains, and so we're here to kind of explore the use case. So that kind of leads me then to the first big problem with data, especially incorporating open data into a workflow where you might also be using proprietary data or self-generated data. So the first thing that you end up is, where are all the data? Can I get it all in one place, right? And we've tried to do a little bit of that using the registry of open data on AWS and with support from the AWS Open Data Sponsorship Program. We try and lower that barrier to getting data into cloud to zero. But that solves just the first piece of the puzzle. The second is, what are the questions I'm going to ask of this data, and therefore how do I structure the data, right, to answer my question, knowing that your question next year may not be the same as the question you're asking today.
And I also hear a lot about, you know, having unstructured data, structured data, but also you might have different fields mapping to different values, or you might have a naming schema that's different.
Totally.
You know, and I understand just with working with a lot of log aggregators, like there's some sort of common information models out there, but it sounds like there's an effort to help make that achievable, but just sometimes the data may come in the way that it was generated, or it's the product of generation.
Exactly, and that's one really good thing to point about the Open Data Sponsorship Program, is that the data that we help to support the distribution of, and the data that are listed in the registry of open data, these are almost always maintained by the data provider. We do have a couple of data sets that live in the public domain that a third party has said, hey, I want to put this data on the cloud, let's do that. And we do even have a couple of data sets that have been produced by Amazon teams.
Oh, that's cool.
But the vast majority of the data are owned and maintained by non-AWS people. So that also means that I can make strong suggestions, but I'm not always going to say, hey, this is how I think you should structure your data, and you're going to do it. Right? Sometimes it's, this is how the data are, and this is what we're going to do to work with it.
Now, I know one thing that you've been saying is make open data better.
Yes.
Could you, what do you mean by that?
Oh my gosh, that's such a good question.
If I had to run around this expo floor saying, make open data better, but here's how would we do that?
Well, I will say that for this specific use case, since we only have 20 minutes today, I will zoom in on the use of purpose-built services that remove the undifferentiated heavy lifting of using public data. And so when we talk about aggregating data, that's step one. The second is structuring the data. The third is actually figuring out what cloud services you need to provision, get them together, get them to work in tandem, so that you can actually ask the question. Because you're a bioinformaticist, right? You might even just be a clinician, or like me, a veterinarian. I didn't know how to code when I started, right, in my profession. So what we want to do is, okay, what is the answer to making open data better? Let's try and funnel it into a service that removes that undifferentiated heavy lifting.
I think you just also hit a fair point too, that, you know, to me as a technical individual, I would just assume up front, oh, technical individuals are using this. But no, we want to extend it to clinicians,
scientists,
healthcare providers that don't necessarily want to understand all the nitty-gritty technical soup behind it. But they want to be able to ask questions to data to make better informed decisions to provide healthcare, veterinary services, and things like that. And so I think that's very important to also understand. And so does that mean that there's tools that they could leverage without needing to know how to use Python or step functions or Lambda functions, et cetera?
Yeah, I mean, it's still going to be helpful, and I will say this myself, right? I did have to kind of learn on the fly. I guess, you know, maybe 10 years ago when we started to leverage the cloud, I was like, oh, I've really got to ramp up quickly, and that's been quite a journey. But that's right. We want to make the power of the cloud to answer important clinical questions available to anybody. Yeah. So that leads us to a service that we actually launched at reInvent last year. It's called Amazon Omics, and it is purpose-built for people asking biological questions.
And
biological being more around the human genome.
It doesn't have to be human only. It can be any organism. In this case, we're going to talk a little bit about human, but you know, we have people who are doing animal studies, who are doing
bacterial studies.
Bacterial, viral.
Did you just tell me aliens are real?
No, though I will say that I have totally gotten inquiries to open data before about alien DNA, and I was like, we don't have that. That I can guarantee we don't have.
That she's willing to divulge.
This I can say. That's true. That's not open.
So you came out with this new service at reInvent. You know, if someone wanted to get hands-on with this service, what are ways that they could go about getting started?
Yeah, so we have a GitHub repository of Amazon Omics tutorials that kind of, I think they lead us through maybe 3 or 4 common use cases of how people are using Omics. And just keep in mind that Omics has a lot of different attributes to it. It's not just, I mean, like, it's not like a monolithic service, right? So there are pieces of Omics that people can use independently of each other or together. So we have things like Omics Sequence Store, which is optimized storage for next generation sequencing data. Like Sujay has said, Omics Sequence Store is huge. It's big data. And so to be able to optimize the storage of that to make cost most efficient, that's going to be super valuable. But we also have things like Omics Variant Store, where the data that come after genomic secondary analysis, where you say, oh, I found a bunch of mutations, right? What do they do? And that's what we're going to focus on a little bit today. Those are also structure optimized to be super performant for query using standard query language or SQL.
Okay.
Yeah. So, I mean, so people in the field even who work with what we call variant call files, and these are sort of domain specific files that, you know, are a specific format, but they are not, you know, great at all if you want to just aggregate a bunch of them and do like querying across.
You can't open them in an Excel file.
Yeah, they're just
too big.
Yeah, they're big, and then the format is just really hard to do anything with. And so you have to do a bit of, you know, quite a bit of transformation to bring it into a form that you can then say, okay, I have now like 10,000 VCFs, and I want to kind of do some querying across them to see, you know, how, like, I want to find variants that are clinically significant, maybe for a specific disease and things like that. And that also brings me to the next point, where the variants only tell a part of the story. There's the biological significance. So the variants will just tell you at this position, there is, you know, there's a mutation that changed from A to C, for example. So it should have been an A, and it's actually a C in your case. Now, what's the significance of that? I mean, a majority of them, nothing. I mean, we're all walking around with lots and lots of these mutations, and there's no effect really. But there are a small subset of those that actually do have some clinical significance. And so, you know, you do have, you know, some diseases are caused by just a single mutation.
Wow.
Yeah.
But also just like thinking about the large data sets itself, just the processing, like we're kind of joking around like, oh, you wouldn't open it up in a spreadsheet program, but really to process these files and the amount of data involved, you would need something that scales to the amount of data being processed, and what better than to use AWS to be able to scale that for you and host that hardware, because having that in your back pocket, it's not really going to happen, right?
Exactly.
Yep.
Exactly.
Yeah.
Sorry, we're trying to keep this live, but in essence, right? So what we wanted to show today, and we do, as you see, our screen sharers may or may not cooperate with us, is the ability to pair genomic variants that actually exist in a real population. It's called the 1,000 Genomes Project, and we want to actually demonstrate that we can use mutations found in that cohort and pair them to regions of clinical significance.
Yeah, let's get into it.
Let's see if we can do it with us.
Okay. So here, actually, so why don't you talk a bit about the 1,000 Genomes open data set?
Oh, absolutely. So 1,000 Genomes is actually one of our our flagship open data sets. I think it's been on the program for longer than I've been with the company. But 1,000 Genome was this endeavor to basically capture the naturally occurring genetic variation across multiple populations in the world. You might not be surprised that in research cohorts there are different ethnicities that are underrepresented, and there are ethnicities that are overrepresented. So 1,000 Genome was really a landmark study that said, hey, actually we need to understand where these genetic variation occur and who they occur in. Right? So that's 1,000 Genomes. So we actually host all of that next generation sequencing data in the Open Data Sponsorship Program. It's readily discoverable. If you go onto the Registry of Open Data, which we've been sharing for a while, and just search 1,000 Genomes, you can recover those data. And the nice thing about those data is that we can use them as a fully public resource to demonstrate.
That's really cool.
Yeah, it is really cool. So that's the background on 1,000 Genomes, and we're actually using that in conjunction with another incredibly useful open data set called DBNSFP. And so this is maintained by a professor in Florida who basically took every public database of known clinical, like that annotate. We call these annotations, right, where you take genetic variants and you tie them to areas of clinical significance, papers, disease
and biological significance.
Yes, he aggregates these data and keeps it up to date.
Wow.
Yeah, he's
so it's
incredible.
like this humongous table of...
Shout out to you. That sounds time consuming.
Yeah. And we also sponsor the storage of that data set.
Well, that's super cool.
Yeah, so these data are living in S3 buckets, but the fact is you then have to say, well, how do I want to structure these data? How am I going to pull these data together to ask questions? And that's where we wanted to show you what Omics can do.
Awesome.
Yeah, so
and so I see Jupyter, so we're working from a Jupyter notebook.
Yeah, so there's a few steps before we got to this point.
It's like a baking show.
Yeah.
That's exactly right. The pie is done.
Yeah. So using Amazon Omics, we actually ingested all of the VCFs from the 1,000 Genomes, as well as DBNSFP. So they're now all tables that can be queried through Athena. So we already have done that part, which in reality is a huge effort if you didn't have Omics. You'd have to write some pretty complex ETL scripts and like process these on like an EMR cluster or something. So it's a pretty big process. But with Amazon Omics, it's a couple of API calls to ingest everything. And now we have these as tables, and what we're showing here is we're able to, if the screen ever comes back on, what we're doing basically is combining the variants, the raw variants from 1,000 Genomes, and joining it with the annotations from DBNSFP, and trying to see how many variants actually exist in BRCA1. Now, BRCA1 is a well-known gene implicated in breast cancer.
Okay.
So that's, we made the whole thing look super easy, but this actually is a pretty involved long process to get here.
And to be honest, I think we actually executed a lot of these steps that would frankly take someone days, if not weeks, to do. I think we did this in an afternoon. We did it actually specifically last Friday afternoon.
Oh, wow.
Yeah, so the fact that we could do this with not a huge amount of overhead was really mind-blowing.
That is awesome. Now, I understand, and this might be a rumor, that you have a workshop here at the DC Summit.
Yes,
that's true.
That will be, was it tomorrow afternoon?
Yes, at 1:30.
So if someone wanted to get hands-on with this,
yes,
they're able to do that here at the DC Summit.
Yep.
Yeah.
Awesome.
That's right.
Yeah.
That's so cool.
Yeah.
Well, we're running up on time, but I wanted to thank you two for one. My mind is blown away just with how much information I just learned about sequencing data and the human genome. Is there anything else you'd like to say to your fans, to the data set folks, or how to make data better?
Well, come to us if you know about valuable open data that you want to make available. We want to help you make it better.
Yep.
Awesome. Thank you so much for your time.
Thank you.
All right, stick around for more Kyle and probably Aaron.